So our next speaker will not be Peter, but will be Christian Beetz, who will talk about the role of the African GBA allele in Parkinson's disease. This is one of two talks about this, so looking forward to hear it.
So also I would like to thank the organizers for the opportunity to present our data, and also the Michael J. Fox Foundation for funding what we are going to present. Starting out with some background, there was this Lancet Neurology paper earlier this year that identified a novel GBA1 PD risk factor and showed it's highly prevalent in sub-Saharan region, and most notably it's an intronic variant, thereby differing from all the known GBA-PD risk factors, and the authors hypothesized that it acts via decreasing GCase activity. When reading the preprint about a year ago, we felt that we can contribute a lot to further understanding the effect of this variant, and that is because we have this huge internal biodata bank, meanwhile composed of more than 250,000 research consented cases, fully sequenced including GBA1, dry blood spot samples available where we can do biochemical assays. And also we have been running this Rostock International Parkinson's Disease Study for more than 4 years now, having enrolled more than 19,000 PD patients and genetically characterized them. So what did we do? We first looked at the whole database, identified all the positives for the African allele, calculated the minor allele frequencies at the country level, and what you see here is a mapping of the maps to the world map for all the countries where we had at least 100 individuals sequenced. So first of all, we clearly confirmed that there is a sub-Saharan hotspot, probably also origin for this variant. Notably, we don't see a second hotspot, but wherever we see the variant, it correlates with the proportion of Black Africans in that country. So we're talking about a single founder event, which may be surprising because the variant very likely resulted from a pseudogene conversion, and such events usually come as mutational hotspots, best known for the L44P variant. We also wanted to understand the effect of the variant on splicing, because we're talking about an intronic variant. So here a scheme of the 11 exon GBA gene, then the canonical transcript, also as input from the lab of Andrew Singleton and Cornelis Brouwendraat. We had learned that they had observed intron 8 sequence to be present in individuals carrying the variant. So we designed a rather simple RT-PCR strategy, 2 PCRs each, conceptually one pair of PCRs targeting regions that are outside the critical region of the GBA gene, a second set of PCRs targeting the critical region but not the intron 8, and finally 2 PCRs that also contain primers in the intron 8. We selected wild-type samples and heterozygous from the biodata bank, extracted RNA and applied these PCRs. So results very consistent across the conceptual pairs of PCRs. When we looked outside the critical region, we didn't see any difference in expression between wild type and heterozygotes. Looking at the critical region, but not considering intron 8, we saw a slight reduction in expression levels, non-significant though. But when we then looked into transcript containing intron 8 sequence, we saw a significant increase in such species in the heterozygous subjects. So concluding that the presence of the African allele does not affect the overall abundance, but rather turns a small, and notably a small proportion of canonical transcript into intron 8 containing transcript. We next asked, how does the intron 8 sequence end up in the transcript? What happens here? And again, based on RT-PCR amplification and then sequencing, here showing the Sanger traces, we determined that there are 2 aberrant events. One is where the whole intron 8 is contained in the transcript, and the other is the major aberrant isoform, actually, where only part of intron 8 is contained. Next, trying to speculate how these observations could be explained, we considered what is necessary for proper intron 8 splicing. So we have the ordinary splice acceptor, splice donor. We also have a branch point which precedes every splice acceptor and is needed for forming these lariat structures that actually represent spliced out introns. And it turns out that the variant falls right into the middle of a predicted branch point motif. Assuming it destroys this motif, our observation of full intron 8 retention is nicely explained. So if you miss a branch point, you can't get rid of the exon, of the intron. Notably, in silico predictions say that there's another nice pair of splice acceptor and branch points somewhere else upstream in the intron. And the consequence would be that you only splice out part of the intron 8, and that exactly explains the second sequence that we observed. But also of note, there's another branch point predicted to be right next to the original one. So the fact that we didn't see a full effect means that we have a lot of normal transcript from the mutant allele as well. And that's very likely explained by the usage of this alternative branch point. So conceptually, we have two effects. One is the partial or full intron 1 retention, leading to truncated and potentially misfolding brown GKs. But we also have a lot of normal GKs usage of an alternative branch point, but resulting in the same transcript in the same normal GKs protein. Finally, we were interested in the effect that the African variant has on GKs activity levels. So we determined these levels from a couple of hundred dried blood spot samples, including non-PD, including non-GBA PD, idiopathic PD, also GBA PD. So first of all, we fully confirmed that GBA PD on average shows slightly reduced GCase activity. But the big question was now, where does the African variant fall in this? And we observed that there is a slight reduction in activity for both heterozygotes and homozygotes, with the homozygotes showing more higher levels of reduction. And most notably, the levels of reduction generally fall within the range of the mildest known risk factors, namely the 365 and the 408 variants. So concluding, we showed two things mainly. So there is evidence for presence of truncated, non-functional, potentially misfolded GCase molecules, arguing or supporting a potential gain-of-function mechanism. But there's also evidence for this being paralleled by fewer functional GCase molecules being present and supported by the lower GCase activity data, thereby supporting a loss of function mechanism. So in that sense, the variant is not special. It resembles all of the other risk factors by not really contributing to a solution of the question that was discussed before. Thank you for your attention.
It's very interesting. Any questions? So I have a quick question. Would you include these carriers now in clinical trials that are happening for GBA?
Would make sense. If you include the 408, if you include the 365, it is enriched in PD. That's all that counts, I would say.
Thank you.